Hello, my dear future doctors. This is Dr. Madonna. Um, this is 3 o'clock of Saturday. No one has joined me yet. I guess because that is because of the time mix-up. But I'm gonna wait a few minutes for you to join me and then get started with today's lecture. Yeah, this is the second lecture of the general and oral pathology. We're at the beginning of the general pathology discussing Robbins chapter 1. Okay, I'll give you a few minutes and get on with my presentation. Okay. Okay, guys. So, in the previous lecture, which was on the Thursday, we discussed the basics of the pathology. We discussed some terms. We talked about the causes of the cell injury. We listed all the reasons, and also we talked about what happens to the cell after the injury. We discussed reversible and irreversible changes caused by the reversible or irreversible cell injury. We discussed the cell death, which was divided into two parts, or two types: the necrosis and apoptosis. And today I'm going to talk a lot more about necrosis, and for the next lecture I will talk about apoptosis. So this one, which I marked with the red color, I'm going to talk on Tuesday. Okay, remember, guys, we have rescheduled our meetings. It will be not on Thursday, but it will be on Tuesday at 2 o'clock. And I'm gonna talk about the rest of the chapter one, which includes apoptosis, cell injury caused by the toxin, DNA damage, inflammation, and the adaptation processes like hyperplasia, hypertrophy, and so on and so forth. Okay, welcome to a tutorial. Just just going. I was getting ready to start. I hope you all have received on your drive the book itself and the syllabus, and the presentation will be uploaded after this lecture, okay? Because I have split this first lecture into two parts. Today I'm gonna finish it, and that's why the presentation you will get after the two lectures, okay? So after today, you're gonna
Okay,
get
thank you, Victor.
PT as well. You're welcome. I hope everything was clear in the previous lecture, but I'm gonna repeat myself a little bit, okay? So, uh, we talked about the introduction of pathology. With introduction started with terminology, obviously. We discussed what the etiology means. Etiology is the Latin word. It's talks about the origin of the pathology itself, origin of the disease, the reason of the disease, why a disease occurs. While pathogenesis is how the disease go on. Okay, how does it develop step by step? Yeah, what the etiology causes is pathogenesis. Okay, so, and we said that each of the cell injury has the morphologic causes morphology changes. Pathogenesis includes morphology changes at the biochemical level, at the molecular level, microscopic level, which some of the changes are not often seen through how to say through without the microscope, light microscopy, or without the special instruments, just by bare eyes. Okay, so those changes are at the morphologic level, molecular, microbiologic, or immunologic techniques are used to define and determine these very changes. Okay, so I talked about what the pathology focuses on. The general pathology, which is the first part of our lecture course, it discusses the alterations that are caused by the pathologic stimuli that occur in the tissues and in the cells. While systemic pathology discussed a bit further, it discussed organs and organ systems that are involved in this pathology and what kind of changes they undergo when the injurious stimuli occurs. Okay. So, when we started, the first thing is to discuss when we're talking about pathology, to discuss the injury the pathology damaging stimulus causes to the cell. The injury can be due to reversible cell injury, or it can be irreversible that results in the cell death. And when we talked about the cell death, we mentioned two types of the cell death. What is predetermined, pre-programmed, it's called apoptosis, and the other one is necrosis. And today I'm going to talk about necrosis a lot more, okay? And for the next lecture, we're going to talk about apoptosis. So these are the injury. What are the follow-ups of the injury in terms of the cell damage? And you can see here the normal cell is usually at homeostasis. It means in balance. Okay, it means the ion exchange is normal, mitochondria are active, they produce ATP, adenosine triphosphate, the energy source in order to utilize it for the cell's well-being, homeostasis. Okay, when the injurious stimuli occurs, whether it is reversible or irreversible, it depends on the stimulus itself, on the duration of this damaging stimuli effect in the cell and the cell's general condition as well. Okay, different cells, different tissues types, they answer to the damage differently. Okay, of course, the duration of the damaging stimuli plays the crucial role. If it's in how to say is it's prolonged and undergoes certain how to say overflows the certain limits, it might cause irreversible changes. Okay, so the dermal cell, which is at homeostasis, is under the how to say permanent stress, but it has tools in order to adapt to these permanent stress. So we've got adaptation for that. If the organism or if the cell is unable to adapt, then we've got the cell injury. Okay. And cell injury could be mild or transient that will cause a reversible injury. It means that the cell can go back to its initial state, maybe not totally go back, but still continue functioning. And when there is no point of return, we've got severe or progressive injury, we've got irreversible damage to the cell that might lead to cell death via necrosis or via apoptosis. Okay, so this is the virtual illustration of the cell injury. This is the normal cell, obviously, and the changes it undergoes when the injury occurs. Okay, so as you can see at this point, we've got the recovery. This is the reversible injury. We've got the recovery of the cell. It means the swelling of the endoplasmic reticulum, mitochondria, membranes, the blebs that could regain its shape and go back to its normal shape. Okay, but if the damage progresses along, we've got the irreversible changes, disruption of the cell membrane, plasma membrane, that interferes with the integrity of the whole cell, and finally we've got the necrosis of the cell, elimination of the cell. And this other way is the, as I said, pre-programmed, planned, not always a bad thing, apoptosis, which is also another type of the death, but less severe, less traumatic for the whole organism, and could be for the benefits of the body itself. Okay, in order to give a place, give a opportunity to the newer, fresher cells to perform their functions better and for the whole organs to function appropriately. Okay, so, and we talk about, I'm not going to stop you, I've discussed it enough, and necrosis. Okay, cell death. Let's get started with the cell death. The cell cannot recover if the cell cannot regain its normal structure and obviously normal function, it dies eventually. So the death again could occur via necrosis, via apoptosis, and the very important feature for the necrosis that differs it from apoptosis is actually a host reaction called inflammation. The inflammation you will not meet during apoptosis, but rather meet it with necrosis. Okay, so cellular contents leak through the damaged plasma membrane, which is the feature of the necrosis, and they happen to become to exist in the extracellular space. That's why they cause the swelling and edema. That are two properties, the characteristics of the host reaction called inflammation. Okay, so necrosis occurs to the various injuries, various damaging stimuli, including ischemia, exposure to toxins, various infections, and trauma as well. Direct exposure to these damaging factors might cause the necrosis of the different types of the tissues they affect on. Okay, and apoptosis is a cell said before that is that could be beneficial for the whole organism and to I'd say perfect its imperfections. Okay, so necrosis is always pathology process. Let's agree on that. And apoptosis serves as a tool for adaptation, for evolution, keeping its role in certain physiological processes as well. And it doesn't elicit an inflammatory response, which is very common during necrosis. Okay. And these are the features of the necrosis and apoptosis separately, the differences between them that will make you to see clearer what is the difference between of these two. Okay, and the causes of the cell injury, we listed this injurious stimulus and discussed each of them. And the difference between hypoxia and ischemia is quite important to understand because as a clinicians, you have to know the difference between these two. So hypoxia, the word is hypo means less, decreased. Oxia, again, oxygen level, so it means the oxygen deficiency. But ischemia is the loss of blood supply. Okay, well, if we imagine, uh, yeah, loss of blood supply cannot survive without the loss of oxygen. Okay, it directly means the loss of oxygen. So ischemia could lead to hypoxia, that could lead to the damages that is caused because of the oxygen deficiency. But not just the oxygen deficiency, but also the nutrients that blood supplies the tissues with. Okay. So most common cause of hypoxia is usually ischemia, and not vice versa. So hypoxia cannot cause ischemia, but ischemia is the reason of the hypoxia. It could be due to um, big amount of the blood loss due to wounds or something else, and due to anemia, due to pneumonia as well, due to oxygen carrying capacity is lost, while like during sickle cell anemia, where they lose their shape and ability to carry the oxygen and other nutrients to the tissues, and also as well carbon monoxide poisoning, which leads to severe damage to the cells. Chemical agents, we said that we live in the industrial era, and each year dozens and dozens of pesticides, insecticides join the, I'd say, join the markets. So they all contain some chemical agents that somehow affect the not just us, the environment as well. The yeah, and microorganisms that live with us. So those chemical agents could have the vicious and deleterious effects on the bodies and our cells as well. Okay, and depends on their again on the duration of the exposure of these chemical agents as well on the overall capacity of the body to adapt to these chemicals to and the overall health that leads to determines how the disease will go on, how the pathology will progress. Okay, pathogenesis in this case. And infectious agents. Infectious agents has always been and will always be neighbors of ours. Okay, so like coronavirus, like other bacterial infections, streptococcus, staphylococcus, fungus, protozoas, and others, many seen or unseen, known or unknown. Those infectious agents definitely have the injurious effects on the body that have the ability to cause different types of disease and changes in the organism. and immunologic reactions. At first, you should think that immunologic reactions are for us to protect ourselves, protect our bodies from foreign. But the immunology reactions sometimes are blind. Okay, sometimes there exist in autoimmune reactions that fight against ourselves, fight against our own tissues and our own cells. And in this case, they could be quite deleterious. And allergic reactions as well against dust, against um, changing seasons. Those also allergic reactions somehow distort the homeostasis of their bodies themselves. Okay, and genetic factors, as in everything, genetics plays a crucial role. Okay, because the predisposition that each of us have is inherited from the parents, from maternal inheritance, paternal inheritance, and so on and so forth, or new mutations might lead to this changes that also this is the thing that differs us from each other. Okay, each individual has a individual pattern of reactions that occurs after the different injuries. Okay, so this is what determines our differences, our different reactions. That's why personalized medicine exists in this in nowadays 21st century is the century of the personalized medicine. What does this mean? This term means that we don't treat the disease only, we treat the disease, we treat the patient. Okay, because each disease has different pathogenesis in a different patient, because a patient is as an individual is unique with its genetic factors, using with its immune system, with its environmental factors. So all this and add up to each other, sum up and make up a how to say epigenetic factors, not just genetic but environmental factors that have a very crucial influence on the pathogenesis of the different type of pathologies. Okay, and another thing we've talked about is nutritional imbalances. It's quite common because yeah, of course, females will understand me well because there are dozens of diets that and even some the popular ones that are somehow advertised in the social media as well, that how to that weak humans tend to follow, but that's not a good thing because the deficiency of the nutrients, deficiency, lack of vitamins, lack of nutritional factors lead to the development of the different types of pathologies. Okay, so that's why nutritional imbalances are one of the causes of the cell injury. So keep that in mind. Everything we eat, we drink, is important for our body. Okay, all the supplements that are used in the reactions that undergo each minute, each second in our body without us knowing about them. So everything is important. You should consider all the benefits, pros and cons, while making this kind of decision, and denying yourself in the pleasure of eating something or drinking something, or vice versa, eating something that you should not and drinking something you shouldn't. Okay, yeah. And the other thing is physical agents. So physical agents, I mean trauma, most commonly is like car accidents. Those are damaging factors. You can see here brain damage, yeah, acceleration, deceleration trauma, toxic agents, extreme temperatures. It doesn't always mean the heat, but also as well cold. Radiation, obviously, ionizing radiation, ultraviolet radiation, that in certain existence of the genetic disorders might cause the might could be cancerous. Okay, cancerogenic, tumorigenic, yeah, mutagenic. And electric shock, obviously, could be even fatal, and sudden changes in atmospheric pressure. yeah, Cassandra disease, and so on and so forth. And last thing is you should not consider it as damaging factor, but obviously it is one of the factors. It's a natural flow of the time and aging of the organism that leads to finally how to say depletion of the energy sources, depletion of the functional functioning itself, and leads to the logical conclusion, which is the cell death. Not logical for our but who knows, maybe in the future we could be immortals and this will no longer be a factor, but still for today it is so. So now let's discuss the morphology of the cell injury. I will increase this one for you to see it better. Okay, here, here we go. Yeah, I should better done this before, but anyway. So all stress and nauseous influences exert their effects first at the molecular or biochemical level. So it means they affect not just the directly at the heart or at the brain, but at the molecular level. It means that the cells of the heart, myocardicytes, the cells of the brain, like example, the neurons, and so on and so forth. So at the smallest level, and they then we go. This is the general pathology we're talking about, and the systemic pathology when we already have effect on the level of the systems and the organs. Okay, cellular functions may be lost long before the cell death occurs. Means that the deterioration of the functioning could be before the actual cell death, before the disintegration of the cell, and the morphologic changes of the cell injury or death like far behind both of them. Okay, so visible changes, not I don't mean with the bare eyes, but with light microscopy with the special instruments and tools, could be even after all of this happens already. Okay, for example, when we discuss the heart tissues, so myocardial cells become non-contractile after 1-2 minutes of ischemia. So ischemia is the loss of blood supply that leads to hypoxia, oxygen deficiency, that leads to myocardial cells to become non-contractile. It means now cardiac cells to lose their contractility, their function. I'd say, okay. So the injury is the ischemia causing hypoxia that causes non-contractility of the myocardial cells, loss of the function in 1-2 minutes. Okay. But how can we detect these changes? We cannot detect these changes in 2 minutes. Also, they do not die until 20 to 30 minutes of ischemia have elapsed. So they lose their function, but they in 1-2 minutes, but they die as physically uh, they disappeared and to die. Necrosis occurs in 20-30 minutes. But this is not the last thing. We cannot see them dying in 20-30 minutes. It takes us 2-3 hours to see them and detect them. Okay, these myocytes may not appear there by electron microscopy for 2-3 hours, or by light microscopy even further, 6 to 12 hours. So that's why we've got. So how can we detect your thinking? How, if we cannot um, see what has already happened 12 hours before, how can we prevent this? We don't see the actual thing that happening, but we have the signs of it to determine, like dropping in levels. To determine these levels, it means decay is already begun, so we act in order to prevent other myocardial cells to die along with that already been dead. Okay, so that is the tool of the medicine, and I know it's a lot, but you will get to it. Okay, so the cellular deterrence of the reversible injury can be corrected, and if the injury stimulates the base, the cell can return to normalcy. If the damaging stimulus stops at a certain level, at the level where the injury is not that serious that disrupts the integrity of the cell, the injury could be reversed. But if the persistent or excessive injury occurs, it causes the cells to pass the point of no return where there is no going back, okay? And it goes into irreversible injury and finally cell death. Okay, and the events that determine when the reversible injury becomes irreversible, it's quite difficult and unknown and is still a closed curtain for us. That is why yeah, that is why we have not much of the tools in order to stop or prevent irreversible changes. Okay, but if chemical molecular changes that predict the cell death can be identified with precision, it may be possible to devise the strategies for preventing the transition from reversible to irreversible injury. So you're asking yourself, so me, that if we don't know these things, why we're studying them? Because you are the fresh minds, okay? You might have a new ideas, and you might be the one that finds this way, finds this clue, and the idea that might seem crazy or not believable now might someday prove to be working. Who knows? So that is the as a benefit of being a student. You are not making mistakes. Okay, what you're thinking, what you're deducting after hearing something is always correct, because yeah, this is the product of your thought, and thinking is the powerful tool and powerful mechanism to evolve and become better. Okay, so go on thinking, and don't be ashamed of your ideas, don't be ashamed of your questions and your thinking. Okay, so let's go on. So there is no definitive morphologies or biochemical correlates of the irreversibility. So this is the obvious fact. So two phenomena consistently characterize irreversibility. This is what we know for sure. The first thing is that inability to correct mitochondrial dysfunction. This is the vital and the crucial. And the second is profound disturbances in the membrane function. Okay, mitochondrial dysfunction I'm going to discuss today. Oh, that leads to damage to the cell that is irreversible, and obviously membrane dysfunction that disrupts the integrity of the whole cell. Okay. So now let's characterize the necrosis. Okay, this is and first let's characterize reversible injury, the properties and my morphologic findings, how we can characterize the reversible injury. It has two morphologic findings: cellular swelling and the fatty change. Okay, and let's discuss each of them separately. First, cellular swell. It results of the failure of energy-dependent ion pumps in the plasma membrane, leading to inability to maintain ionic and fluid homeostasis. It means the contents of the cell start to leak outside into the extracellular space, and the cell starts to swell. Okay, so this is the first feature of the reversible injury. Okay, injury but still could be reversed, not the fatal one. And the second thing is the fatty change. What does that mean? It occurs in hypoxic injury, oxygen deficiency injury, and in various forms of the toxic and the metabolic injury as well. And what is the manifestation of the fatty change? It appears as small or large lipid vacuoles in the cytoplasm. Okay, lipid vacuole fatty change, so they're connected. They appear in the cytoplasm. It is the sign of still reversible injury, but an injury. And now let's go on. So potentially injurious insults induce specific alterations in the cellular organelles like endoplasmic reticulum. ER is not emergency, but is endoplasmic reticulum, which is the place where the ribosomes reside, where the place of translation, and yeah, yeah, the very important place, and organelle, obviously. And the smooth endoplasmic we've got the rough, where because it's called with the ribosomes, it's called rough, and the smooth without ribosomes, and it has flat cisterns. I'm just reminding you the 12th grade cell biology, so you should know that. The smooth endoplasmic reticulum is involved in the metabolism of the various chemicals. That's why it's called detoxicating organelle. So, and the cells exposed to these chemicals show hypertrophy. Hyper means much, hypo less, okay, decrease and increase. Okay. Hypertrophy, it means enlargement, okay, enlargement of the endoplasmic reticulum as an adaptive response that may have important functional consequences. So it enlarges in order to perform its function better. It's an adaptive tool. And when we next lecture on Tuesday, we're going to talk about adaptive reactions, and hypertrophy is one of them until it becomes decompensating, obviously. And for example, here we have a good example: barbiturates. Those are the drugs used during epilepsy, anti-seizure drugs, sedatives. So barbiturates are metabolized. You will learn them in pharmacology, or you will have discussed them. I don't know. Okay. Barbiturates are metabolized in the liver. Liver is the place where the most of the drugs are metabolized, because there's activity of the cytochrome P450 system, and these barbiturates are metabolized by this system, the mixed function oxidase system found in the smooth endoplasmic reticulum of the hepatocytes. Hepatos means liver, so liver cells, hepatocytes. Okay, so this is the cytochrome P450. This is digital view of it. And a protracted use of barbiturates leads to a state of tolerance. Tolerance, what does it mean? It's a decrease in the effect of the drug. And in order to achieve the wanted effects, we need to increase the dose. Okay, so it's no longer the previous dose is effective, but in order to achieve the effect we had at the low dose, we need to increase the low. So the tolerance accused to these drugs, and eventually might lose its property at some point. Okay, but it's rare. This adaptation is due to, so this is an adaptation. Tolerance is a tool as well. So this adaptation is due to increased volume, hypertrophy. Hypertrophy of the smooth endoplasmic reticulum of the hepatocytes or liver cells, and consequent increase of the P450 enzymatic activity. So, as the size of hepatocyte smooth endoplasmic reticulum increases, also it increased the cytochrome activities as well. It means the metabolism as well, metabolic, so the rate of metabolism of the barbiturates as well increases. It means the tolerance develops quickly. And P450 mediated modifications is often thought to be as detoxification. It means the tolerate we tolerate to these drugs, and so many compounds are rendered more injurious by this process. Okay, so as we need higher dose in order to get in the effect, so they become detoxified, but the drugs become more injurious. Okay, because the high dose, high side effects, um, frequent side effects. Okay. And the other thing is carbon tetrachloride. Tetra means four, as you can see, C chlor 4. The products formed by this oxidative metabolism include reactive oxygen species. ROS. Remember, ROS, not red, but the reactive oxygen species. Those are injurious, okay, like the free radicals ones. So those are injurious to the cell. And the cell adapted to one drug have increased capacity to metabolize other compounds handled by the same system. So as I said, barbiturates are not the only drugs that are metabolized and delivered by cytochrome P450 system, but the other drugs. So it means if the tolerance developed to the one of them, the tolerance might extend to other the groups of the drugs. Okay. If patients taking phenobarbital, it's barbiturate, obviously, for epilepsy, increases their alcohol intake. Alcohol is also metabolized in the liver, you know that well. So they may experience a drop in the blood concentration of the anti-seizure medication because toleration to the alcohol means the tolerance to the barbiturates. So anti-seizure medication concentration lowers and the patient is at risk to develop the seizure. Okay, so the reason we were given this drug will appear again because the dosage will not be enough because of the tolerance. So the anti-seizure medication at the subtherapeutic levels because of induction of the smooth endoplasmic reticulum in the response to the alcohol. Okay, so alcohol might decrease the seizure threshold. Okay, and the anti-seizure effect of the phenobarbital in this case. Okay, so, do you understand pharmacology at the molecular level? This one is called. So now for a lot more about the cellular swelling and the fatty change, with which I said those are they are still reversible changes, but injuries. Okay, and the signs of the injuries. So the cellular swelling. The first manifestation of almost all forms of the injury to the cells is a reversible alteration that may be difficult to appreciate with the light microscope, but it may be more apparent at the level of the whole organ. So swelling is quite apparent when we see edema with the bare eyes, but sometimes at the light microscope levels it will be difficult to detect. Okay, when it affects many cells of the organ, it causes some pallor as a result of the compression of the capillaries, increased turgor is the tension, okay, tension of the as well, and increase in the weight of the organ. So liver swelling or the spleen swelling during the trauma, splenomegaly, hepatomegaly. Though that's quite apparent. And the microscopic examination may reveal small, clear vacuoles within the cytoplasm, and these represent distended and pinched off segments of the endoplasmic reticulum. And this is the pattern of the non-lethal injury, again, not irreversible, but non-lethal injury that is sometimes called hydrophic change or vacuolar degeneration. Okay, so cellular swelling, hydrophic change, vacuolar degeneration. Now, what about fatty change? As I said, this is the appearance of the lipid vacuoles in the cytoplasm and usually happens in the cells which participate in the fat metabolism itself, like the cells of the liver, hepatocytes, the cells of the heart, myocardial cells, and it is reversible just like cellular swelling. But it is easier detectable because of increased use of eosinophilic staining when we do hematology eosinophilic staining. Okay, so now these were all reversible things. So intracellular changes associated with reversible injury again. The first thing is the plasma membrane alterations such as blebbing, blunting or distortion of the microvilli, small villi outside the plasma membrane, or the loosening of the intercellular attachments. Those are still reversible changes, but changes that occur in the plasma membrane. So you can see here. This is this is okay. This is irreversible thing. Mitochondrial changes such as swelling and appearance of the phospholipid-rich amorphous densities, those are still reversible injuries. And the dilation of the enlargement and the hypertrophy of the endoplasmic reticulum with the detachment of the ribosomes. Detachment, it means they become rough, become smooth, and dissociation of the polysomes, it means polysomes are many ribosomes together, which are crucial and essential for the translation process. Translation process, when I'm saying it means protein synthesis, okay? Protein synthesis stops proteins that are involved in the vital functioning, also cell deteriorates, okay? And when we've got the nuclear alterations with the clumping of the chromatin, okay, not karyopyknosis or karyolysis or karyorefrexis, but we've got clumping of the chromatin, which still does not mean irreversible injury. And the cytoplasm may contain phospholipid masses that are called myelin figures. Myelin figures are the features, morphologic features of the reversible injury. These are derived from the damaged cellular membranes. Okay. Okay. And this is the morphologic picture of reversible, normal, reversible, and irreversible change. That I've taken out this illustration out of your book. You can check it out there. So this is the normal kidney tubules. You can see this is the tubules with the epithelial cells. These are the nucleuses of the cells. These are tubules aligned beautifully. You can see this intracellular, extracellular spaces beautifully. And the second picture is the not still reversible change. You can see here early reversible ischemic injury. It means the loss of blood supply. The surface is bleb, not as smooth and in the normal picture. And an increased eosinophilia. It means the color of you can see a lot more, and the swelling is occurred. You can see they're no longer smooth, but the blabbing and the swelling occurs, hypertrophy of the cells. And lastly, this is totally different picture. You can see the necrotic, which is irreversible change that occurs. Here are still the nucleuses. Here still you can see there are nucleuses have disappeared already. It means that they lose of the nuclear piece and they are fragmented. So fragmentation of the cells occurs and the definitely leakage of the contents. No insides are left. They are all leaked outside into the extracellular matrix. Okay, this is quite detectable thing. So I will leave you with this slide on and I'll be back in 10 minutes, okay? Okay.
Okay, thank you, doctor. Thank you so much.
Thank you for being here. Thank you very much. Did you find the book and the syllabus on the drive?
Okay, wait. But I want to ask you,
Yeah, please go ahead.
uh, who's the best of I can study book or lecture online?
Book you should definitely read. It's not a big but chapter one is just 29 pages, but if you read the book and rewatch the lectures and ppts, the ppts will help you to review or and to make this online
Ah,
what you
okay.
had. Yeah.
Okay.
But the book, definitely, you cannot skip the book, okay?
Ah,
Because
okay.
more of the questions will be from the book directly, so it will be easier for you to do the quizzes when you have already read the book, okay? So I
Yeah,
advise you like this.
it's a same idea. Okay,
Mhm.
thank you, doctor.
Thank you, thank you. See you in 10 minutes.
Okay.
Welcome back to the tutorial. It's been quite a big pause, but the student was calling me about there were some problems with the insurance. They sent the note that sorry, they sent it a note. they they will suspend the student who hasn't paid for the insurance. I don't know if it's true or if it concerns all of the students. I hope it's not because it's quite a big price to pay. And oh, okay. Okay, let's go on then with the necrosis and so on and so forth. Okay, we've got half an hour still ahead of us. Okay, so necrosis. Necrosis is the one type of the cell death that is irreversible. So, what are the properties or characteristics of the necrosis? So, this is the type of the cell death that is associated with the loss of the membrane integrity and the leakage of the cellular contents culminating in the dissolution of the cells itself. Largely it results from the degenerative or degradative actions of the enzymes and the lethally injured cells. Okay, so the disruption of integrity of the membrane, the leakage of the contents, and the disruption of the cell itself. Okay, so this is the necrosis and the inflammatory response, which is the part of necrosis itself. So the leaked and the cellular contents often elicit a local host reaction, which is called inflammation, and that attempts to eliminate the dead cells and to start the subsequent repair process. So it's the one try of the cell to regain its life and to regain its function, but fails, and eventually the cell undergoes necrosis. So the enzymes responsible for the digestion of the cell may be derived from the lysosomes of the dying cells themselves and from the lysosomes of the leukocytes that are recruited as the part of the inflammatory reaction of the dead cells. Okay, so I think it's quite clear. So necrosis. What are the cytoplasmic changes that the cell undergoes during necrosis? The first, necrotic cells, they show increased eosinophilia. What does that mean? It's pink staining from the eosin dye. Hematoxylin eosin, you know that. So the HEE letter is from eosin. So eosinophilia, pink staining increases when the necrosis occurs. Attributed in part of increased binding to eosin to the denaturated cytoplasmic proteins, and in a part of the loss of the basophilia. So increase of eosinophilia, loss of the basophilia. Basophilia is the part of blue or part of H in hematoxylin eosin part. Okay. Compared with the viable cells or the living actual healthy cells, the cell may have a more glassy or homogeneous appearance, though the one kind of appearance, glassy appearance, and mostly because of the loss of the glycogen particles, loss of intermembrane integrity, leakage, and all of that. So the rest of the changes include myelin figures. Myelin figures first were mentioned when we talk about reversible injuries, but here myelin figures are more prominent in necrotic cells than during reversible injury. Okay, if we compare the reversible and reversible, myelin figures are more prominent here. When enzymes have digested cytoplasmic organelles, the cytoplasm becomes vacuolated and appears moth-eaten. So So what does it? You can see the pictures and the illustrations in your book, please. And so it means the digestive cytoplasmic organelles, vacuolated and moth-eaten appearance they have. This continues in the plasma and organelles membranes, marked dilation of the mitochondria with appearance of the large amorphous densities, disruption of the lysosomes, and intracytoplasmic myelin figures. All this listed above are characteristic post-cytoplasmic changes of the necrosis. You don't have to memorize all of them, just what you can, okay? So this is all the changes that appeared in the cytoplasm. Now, what happens in the nucleus? The nucleus is the core of the cell, the center of the cell, where the genetic information lays in. So the DNA is located. So or RNA. So nuclear changes are important in necrosis because they differ from the other types of cell death like apoptosis, okay, or autophagocytosis, and so on. So the first thing that happens is due to breakdown of the DNA and the chromatin. We've got the basophilia of the chromatin may fade, disappear. Okay, it's called karyolysis. Karyolysis. This is the presumably secondary to deoxyribonuclease activity. So DNA breaks down the chromatin, and karyolysis happens. And the second pattern in is pycnosis. Pycnosis is the nuclear shrinkage, and you can see here basophilia disappears. It means cardiolysis happens, and pycnosis is the shrinkage of the nucleus and increased basophilia because it's darker here, and then here is lighter, and the DNA condenses into the solid shrunken mass, as you can see on the second picture. And lastly, in the third pattern, here we have karyopyresis. It's the breaking down, burst. The pycnotic nucleus undergoes fragmentation. Okay, so it's all fragmented here. And lastly, we don't see any nucleus here, so it disappears. So the last thing, in 1 to 2 days, the nucleus in a dead cell may completely disappear. So loss of the nucleus is the apparent sign of the necrosis. Okay, that was a beautiful entrance and leaving the stage. So, what is the fate of the necrotic cells? The cell may persist for some time without the nucleus, obviously, or may be digested by enzymes and disappear totally. Dead cells may be replaced by the myelin figures, which are more prominent with the necrosis and then with the reversible injuries, as I said before. And this fatty acid binds calcium salts, which may result in the dead cell ultimately become calcified. So they are like monomerized, or they are like cementified with the calcium salts, okay, the death cells, I mean. So let's sum up what we said before in this PPT. So morphologic alterations in injured cells and tissues include reversible cell injury and irreversible, which includes necrosis and apoptosis, and just necrosis we've discussed so far. So reversible cell injury is cell swelling, fatty change, plasma membrane blebbing or the loss of microvilli, mitochondrial swelling, dilation of the endoplasmic reticulum, eosinophilia due to decreased cytoplasmic RNA. So those are the properties of the reversible cell injury. Now, let's talk about the necrosis. Necrosis is equal to increased eosinophilia, decreased basophilia, nuclear shrinkage. We've got fragmentation of the nucleus, and lastly, dissolution of the nucleus. Karyopyknosis, karyolysis, karyopyknosis, karyorexis. and breakdown of the plasma membrane, organelle membranes as well, abundant myelin figures, prominent for necrosis, leakage of the contents, and the enzymatic digestion of the cellular contents lastly. So this is all that sums up what we discussed so far. Now, let's talk. This is actually things that I'm not going to talk, and this is your homework. You should read the morphology of different types of necrosis that is listed in the orange caps, orange boxes of your books. There are 5 types of necrosis with a few abstracts discussed. You don't have to memorize all of them, just remember what you get. So, coagulative necrosis, liquefactive necrosis, gangrenous, caseous necrosis, and the fat necrosis. There's 5 of them. It's just discussed in a single abstract, so I advise you to read it because in the quiz you're gonna have questions about them, and I'm not talking about them, just listing them. Okay. So now let's talk about the mechanisms of the cell injury. Today I'm not going to talk about all of the mechanisms, but briefly discuss the oxidative stress and so on. So the cellular response to the injurious stimuli depends on the type of injury itself. It depends on the duration of the injury, how long it affects the cell, and its severity, how strong the stimuli itself is. Okay, and also depends the the general condition of the organism, general predisposition of the organism as well. So low doses of the toxin or a brief duration of ischemia may lead to reversible cell injury. It means the changes are still could be reversed, okay? Whereas the larger toxin doses or a longer duration of the effects or ischemic intervals, they may result in irreversible injury and finally the cell death, which there is no turning back from. Okay, so the so it means the different cells have different capacities, different tissues have different abilities to adjust to the injury, and consequences of an injurious stimulus depends on the types of the cell, as I said, on the status of the cell and adaptability and the genetic makeup of the injured cell, okay, or whether they have the ability to adapt, to replicate or regenerate or so on and so forth. And the same injury has vastly different outcomes depending on the cell type and not just cell, I would say more on the organism itself. Okay. So here we have different type of skeletal muscle and the different type of muscle cells. Here we get cardio muscle, skeletal muscle, smooth muscle, different locations obviously. So striated skeletal muscles in the legs, for example, here we have in the arms, and the legs they accommodate complete ischemia as a total ischemia, loss of the blood supply for about 2-3 hours without irreversible injury. It means that we cut off not we, if the accident happens and someone cut off his leg or arm, and cut off means that it has no blood supply, so the muscles and the tissues muscles can survive 2-3 hours without the total loss of with the total loss of the blood supply. So it means after 2-3 hours we would be able to rejoin the muscles together and they will still regenerate and they perform their function. Not the same as before, but most likely after the rehabilitation course, the same. Okay, but if the same thing happens to the heart, what will happen? They cannot. The cardiomyocytes, cardiomuscle cells cannot live 2-3 hours without the oxygen, without the nutrients. Okay, so cardiac muscle dies after 20 to 30 minutes, as I have mentioned before. They die in 20-30 minutes, and even less is required for the brain cells to die. 6 minutes, 7 minutes is the top that the neurons cannot survive hypoxia. Okay, so this is the great gives you a great idea how different types of cells react to the duration of the injury. Okay, so mechanism of the cell injury. This is my beloved brain. So the nutritional hormonal status can also be important when we talk about the mechanism of the cell injury. Hepatocytes or the liver cells will tolerate ischemia much better than one that has just burned its last glucose molecule. So hepatocytes have the deposits of the glucose, so they can tolerate the glucose deficiency because they can burn the one they have stored before. But the brain does not have any storage. It directly pumps the glucose out of the blood, and when there is no supply, there is no glucose. It just dies. It has no resources, no other resources to survive. Okay, the genetically determined diversity and metabolic pathways can contribute to differences in the responses to injurious stimuli. And when exposed to the same dose of the toxin, individuals who inherit variants in genes encoding cytochrome P450 may catabolize the toxin at a different rates, leading to the different outcomes. That's quite a logical conclusion because different individuals have different buildup of the liver, buildup of the cytochrome P450 that have different rates of metabolism, and it means that different toxic injurious effects have a different outcomes on a different organisms. Okay, so the principal targets of the biochemical and mechanisms of the cell injury are what mitochondria, because and the ability to generate ATP, and the reactive oxygen species. ROS is under reactive oxygen species. Okay, so the first target of the mm, uh, injury is the mitochondria, because they are a source of the energy, like they generate ATP out of nowhere, and also reactive oxygen species that lead to many pathologic consequences after that. So cell injury from functional to biochemical abnormalities and one or more several essential cellular components, and here are the following. The second thing they affect is the disturbance of the calcium homeostasis. So ATP depletion is the first thing. Then we've got free radical or oxidative stress. Then we've got calcium homeostasis disturbance, because the calcium efflux can uh, cause a lot of damage to the cell. Uh, sorry, the damage to the cellular membranes. Okay, plasma membrane could be lysosomal membrane in case. Then the contents of the cells happen to be outside, like lysosomal enzymes, and they digest the cell. Okay, and the damage of the DNA, and as well misfolding of the proteins. DNA damage, replication damage, misfolding of the proteins leads to formation of the non-functional proteins, and functional performed leads to deleterious effects. And now let's get started with the first, the depletion of the ATP. So as I said, ATP is ultimate source of energy. It's easily formed, easily gained, and easily wasted as well. So ATP is the energy store of the cells and is produced mainly by oxidative phosphorylation of the adenosine diphosphate (ADP). Adenosine D means two, three means T means three. Okay, during reduction of the oxygen in the electron transport system of the mitochondria. So I'm not stopping here because you, the chemistry and all the things you've studied ahead, you will help you to understand this. So in addition, we've got the glycolytic pathway, which can generate ATP in the absence of the oxygen using glucose derived either from the circulation or from the hydrolysis of the intracellular glycogen. So ATP, we know where we get it, how we get it, and depletion of ATP might be the major cause of the reduced supply of the oxygen itself and the oxygen and nutrients as well. Mitochondrial damage, which mitochondria are the sources of the places where the ATP becomes ADP becomes ATP, and the actions of the some toxins like cyanide, poisons, toxins that cause the depletion of the ATP. And tissues with a greater glycolytic capacity, for example, like hepatocytes or the liver cells, they're able to survive the loss of oxygen and the decreased oxidative phosphorylation better than the tissues that have limited capacity for the glycolysis. As I mentioned before, like the brain, okay, they don't have the actual, they just can get the glucose, okay, the energy from ATP, no, from glucose. They don't have a supply or the storage or extra backup plan, okay, like hepatocytes do. So that's why the hepatocytes survive longer with the environment depleted with the ATP, okay. In total, the cells of healthy human burn 50 to 75 kilograms of ATP per day. Okay, every day we use up 50 to 75 kilograms of the ATP. Okay, it's the average a human can use. Okay. So now the significant depletion of ATP has a widespread effect on the many critical cellular systems, including the mitochondrial damage and so on and so forth. The activity of the plasma membrane ATP dependent sodium pumps is reduced, obviously, because of the lack of the ATP, resulting in the intracellular accumulation of the sodium and the efflux of potassium. So iron and this balance occurs in here, and the net gain of the solute is accompanied by the isosmotic gain of the water, causing the swelling of the cell and the dilation of the endoplasmic reticulum. So, what are these signs? Dilation of the endoplasmic reticulum, swelling of the cell. This is the reversible change, right? Okay. So there is a compensatory increase in anaerobic glycolysis in an attempt to maintain the cell energy sources. As a consequence, intracellular glycogen stores are depleted rapidly and the lactic acid accumulates. So this also lactic acid accumulation is additional stress to the cell. So leading to the decrease in intracellular pH and pH decreases, acidic environment, we've got some enzyme activities change. So as a result, we've got decreased activity of many cellular enzymes by metabolism of the cell decreases, and finally we've got the irreversible changes. Here we have mitochondrial damage that leads to depletion of the ATP, multiple downstream effects, and mitochondrial damage can also lead to increase of the reactive oxygen species like the damage of the lipids, proteins, DNA, and so on and so forth. Now, the other things that it caused the depletion of ATP causes is the failure of the ATP dependent calcium pumps that leads to the influx of the calcium, okay, calcium ions. So calcium is essential for the um, function in many of the cells. Like myocytes cannot contract with the calcium, but excess calcium might have the deleterious effect, damaging effect on the numerous cellular components. Okay, so prolonged or worsening depletion of ATP causes structural disruption of the protein synthetic apparatus, manifested as detachment of the ribosomes from the rough plasmic reticulum, becomes smooth. In, the course of irony, and the dissociation of the polysomes into monosomes, no longer form the protein synthesis, and with a consequent reduction of the protein synthesis, obviously. So mitochondrial permeability increases when the calcium entry increases, and the activation of multiple cellular enzymes which digest the cells. Okay, there is irreversible damage to mitochondrial lysosomal membranes, and the cell undergoes necrosis. So necrosis because of the calcium entry too much. So another thing that happens, that was when mitochondria happens, when calcium influx happens. Now what happens to the membrane and the proteins and DNA? So there is irreversible damage to mitochondrial lysosome membranes and undergoes necrosis. This is what I said. Uh, I repeated myself. But what happens to the membrane? Plasma membrane integrity disrupts because ion disbalance occurs. It loses the cellular components, obviously. And lysosomal membrane, what it happens? Enzymatic digestion of the cellular components because the lysosomal membrane disrupts, contents of the lysosomes happen to be in the cytoplasm already outside, and they start to digest the cell and the wall. When we talked about the lysosome, we know that the lysosomal enzymes are active only at certain pH, but we know at the normal pH they are unharm, unharmful. They cannot be harmful to the cell. But in here we've got the pH as well decreases because of the influx of the ions. Okay, so at this pH, enzymes are active and they degrade the cell itself. So protein misfolding and the DNA damage happens because again polysomes dissociate, become monosomes, and no longer protein synthesis is just as it was, and activation of pro-apoptotic proteins happens. That is what happens. So if we sum up all this, ischemia first strikes the mitochondrial apparatus. Mitochondria. Then we've got oxidative phosphorylation decreases. We've got ATP depletion, and if the ATP decreases, we've got natrium pump decreasing. It causes influx of the calcium, water, and the natrium, and efflux of the potassium. As a result, we've got the swelling of the endoplasmic reticulum, cellular swelling, and loss of the microvilli flaps. A second thing is aerobic glucosylases increases in order to compensate for the loss of the source of the energy. But at least we've got depletion of it. We got lactic acid accumulates, glycogen decreases, pH decreases because of the lactic acid, obviously, and the clumping of the nuclear chromatin happens, and also lysosomal enzymes become active on this decreased pH, and detachment of the ribosomes happen from the roughened and plasmic reticulum. Polysomes become monosomes, and no longer protein synthesis can be performed. So this is all the vicious effects that the ischemia and the ATP depletion might cause to the cell. Okay. Now, mitochondrial damage and dysfunction. Dysfunction of the mitochondria. Mitochondria are very sensitive organelles, very important but sensitive organelles to many types of dangerous stimuli, which includes hypoxia, oxygen deficiency, chemical toxins like cyanide, and radiation as well. And it causes failure of oxidative phosphorylation, which was in the previous slide, leads to progressive depletion of ATP, culminating in the necrosis of the cell. Abnormal oxidative phosphorylation leads to formation of reactive oxygen species, ROS, and which may have deleterious effects, obviously. And the damage to mitochondria is often associated with, okay, I will make this red. Ross is means red. And damage to the mitochondria is often associated with the formation of the high condensate channels in the mitochondrial membrane called mitochondrial permeability transition pores that lead to the pH changes and compromising oxidative phosphorylation, and the cycle becomes vicious. And the mitochondria also contains several proteins that release into cytoplasm tell the cell there is internal injury, so there is some kind of uh, emergency siren, and the pathway of apoptosis can be initiated. So not just necrosis, but as well apoptosis is initiated because of the mitochondrial damage and dysfunction. Okay, this is what hypoxia or the toxins and radiation might cause the mitochondrial damage. Mitochondrial damage with the inflammation, this reddens inflammation that leads to necrosis. So mitochondrial damage of the dysfunction leads to ATP. Generation decreases, ATP depletion, production of the reactive oxygen species that leads to multiple cellular abnormalities, and finally necrosis. But if we have, for example, like survival signals from DNA protein damage is detected, it shares internal signals that there is an injury inside the cell. So it activates pro-apoptotic proteins and deactivates anti-apoptotic proteins. And plus, we do have no inflammation, no necrosis, but we've got apoptosis, leakage of the mitochondrial proteins as well. Now, so we talked about depletion of the ATP, we talked about mitochondrial damage, and now what happens to the when the influx of calcium? Calcium, as I said, is crucial ion for the functioning of the many tissues, but excessive calcium can lead to deleterious effects. Ischemia and certain toxins can increase cytosolic calcium concentration, initially because of the release of the calcium from the intracellular stores. And later, it results from the increased influx across the plasma membrane because there is no integrity of the plasma membrane. Increased cytosolic calcium activates a number of enzymes with a potentially deleterious cellular effects, and that's the calcium exerts its deleterious effects on the cell. These enzymes include phospholipases. As you know, ases means the breaking down. Phospholipases break down phospholipases. They cause the membrane damage. Proteases, they break down both the membrane as well as cytoskeleton proteins. So protein breakdown. And the nucleases, as you can say, they break down DNA and they cause the chromatin fragmentation. And lastly, ATPases, which also break down ATP adenosine triphosphate and faster cause the deeper and severe depletion of the ATP. Induced apoptosis by direct activation of the caspases. Caspase is one of the proteins, one of the enzymes involved in the apoptosis. When we're going to mention them, when we're going to talk about apoptosis, and by increasing mitochondrial permeability, permeability against the yeah ions. So this is all what I've talked about, illustrated in with words, with pictures. Okay, so we got the influx of the not influx, but an influx of the calcium because of the effects of injurious agents, toxins, and so on. Excessive calcium causes mitochondrial damage, the swelling of the smooth endoplasmic reticulum, increased cytosolic calcium. It activates cellular enzymes, which I've listed before. Phospholipases break down phospholipids and membranes. Proteases disrupt the membrane proteins, the cytoskeleton proteins, so membrane damage again. And the nucleases, which disrupt DNA and the nuclear damage. ATPase, which cause the further depletion of the ATP and mitochondrial permeability transition as well. So this is all the deleterious effects of the calcium. And now oxidative stress. With a few words, I will talk about the accumulation of the oxygen-derived free radicals, and it's called oxidative stress. Okay. So, the single unpaired electron in outer orbit. So, the free radicals, oxygen-derived free radicals, are the ones that have a free single unpaired electron on their outer orbital. Okay, and such chemical states are extremely unstable. They tend to make bonds with the other radicals. Okay, free radicals readily react with inorganic or organic chemicals as well, and then they attack nucleic acids and as well as the variety of the cellular proteins and lipids and disrupt their integrity. and initiate the reactions in which molecules that react with free radicals are themselves converted into radicals, or the types of free radicals, and it starts the chain of the damage. So it's a vicious cycle. Radicals react with other inorganic chemicals, and I'd say they push the process of them converting into other types of radicals, and it's go on, and it starts the oxidative chain and oxidative stress. Okay, so the reactive oxygen species, as I said, ROS. What does it mean? Uh, ROS. It's ischemia, reperfusion, chemical and radiation injury, toxicity from oxygen and other gases, cellular aging, microbial killing by phagocytic cells, and tissue injury. They all caused by inflammatory cells. Okay, and there are different types of the reactive oxygen species, and they produced in two major pathways, which you can see in this AB picture that I took out from your book. This is the mitochondria, obviously. That's the easiest and fastest way to produce radicals, mitochondria. So here we got oxygen. Here we have superoxide of the oxygen, and it is less likely to leave the mitochondria because it's not permeable. Okay, but it becomes hydrogen peroxide H2O2, which can easily leave the mitochondria and happen to be in the cytoplasm, and sodium. Uh, okay, do this be smooth, or this is something I should remember. So this is the bifenton reaction. So is hydrogen peroxide in the presence of the metals like ferrum, for example, like iron, becomes highly active hydroxide. Okay, this one. So this is called Fenton reaction by chemistry. Yeah, class one. Yeah, this is how the free radicals in the mitochondria form. This is one pathway. The other is then the phagosome. Okay, phagosome, the phagoside oxidase. Here we have net pH against the peroxide of oxygen, and here we have oxychloride, um, hydroxychloride, and this is also as well one of the formation weights of the reactive radicals. So we've got redox reactions, it means reduction oxidation reactions, and respiratory burst. So this is the respiratory burst redox reaction. Now, the damage caused by free radicals is determined by the rates of the production and the removal of what? Okay, so this is the radiation toxin reperfusion reactions, for example, the removal of the radical, how it happens by the way of, um, sorry. Okay, this is again the scheme from your book that you're gonna see in your book and read it. Okay. Okay. And reactive oxygen species, they can cause cell injury by three main reactions. What are these three main reactions? First, lipid peroxidation of the membranes. They double bonds in the membrane. Polyunsaturated lipids are vulnerable to attack by oxygen-derived free radicals. And the second thing is cross-linking and other changes in proteins that is the free radicals promote cross-linking, resulting in uh, enhanced activity of some enzymes that degrade enhance degradation, and free radical reactions that may directly cause the polypeptide fragmentation. Okay, and lastly, DNA damage. The free radical reactions with the thymine and the nuclear and then mitochondrial DNA, they produce single strand breaks, such as DNA, the such like example of the DNA damage has been implicated in the cell death, aging as well, or and malignant transformation of the cells. Okay. Thank you very much. Yeah, that's my advice to you. Please use your really good brains. Thank you very much for your attention, for your attendance, which is 1% today. But thank you still. I hope you're gonna watch the presentation afterwards, and I'm gonna send it to you as well. Okay. Thank you very much. Wish you a good day, wish you a good weekend, and uh, a good week ahead of you. Okay, we're gonna meet each other on Tuesday at 2 o'clock uh, from work. We meet each other from work, and I hope to see you all there. And then, bye bye. Have a nice day. Have a nice weekend, and stay healthy. Stay home, and stay safe. Bye bye.